wherever you are in the world, because I do see that we have people from India even attending. So indeed, as you have noticed, well, this is being recorded, and I will be coming back to this in a minute. But first of all, I would like to welcome you and to set a little bit the scene. So this is the final webinar in a series of 6 webinars that we organized on behalf of the EUROTOX specialty section on in vitro and in silico toxicology, or shortly In2Tox. I hope you're familiar with In2Tox, and I even hope more that you're familiar with EUROTOX. If not, please go to the website of EUROTOX. You will find all about us, also our drivers, what we actually want to do with the specialty section. I also want to cordially welcome you already for the meeting that we will be having, actually an in-person meeting for a change that will take place in Maastricht as part of the combined ICT EUROTOX meeting. So will take place in September. We will have, like all of the other specialty sections within EUROTOX, I think a 1-hour time slot where we will be giving you an overview of our activities, and we also will present our activities for next year, which are pretty exciting actually. So it's a bit of a teaser, and I hope to welcome you there. So as mentioned, well, we try to be quite active in In2Tox, and of course our hands were still a bit tied over the last year, even the last half year, because of the COVID situation, and that's why instead of having an in-person activity, we opted to have this series of webinars. We had already 5 of them. As you can imagine, of course, we focus on topics that are relevant to In2Tox, so either in vitro or in silico toxicology, or even both. Can I please ask you to mute yourself, please? There's a lot of background. Yeah, thank you so much. Actually, this brings me to the housekeeping information I wanted to share with you. So as mentioned, indeed, this is, hello. This is being recorded, so for those who would like to watch again after this webinar, or for those maybe colleagues of yours that were not able to attend, it will be uploaded to the website, the EUROTOX website. Can I please also ask you to mute yourself, because somebody is really shouting now, so it's a bit annoying, and probably it will also be for the speaker. So please mute yourself, please switch off your camera. Of course, we like this to be an interactive meeting, but in order to, not make it too chaotic, I would suggest that during the presentation, you share or ask your questions via the chat. I will be following up on this, and after the presentation, you will get the chance to either speak up, or if you're a bit shy, then I will read your question out loud, and then the speaker will answer your question. Now with this, it's my great honor to introduce the speaker of today, who is Markus Frericks. He has a background in biology and holds a PhD degree that was focused on aryl hydrocarbon receptor biology. He's currently the co-chair of the German Toxicology Society Specialty Section on Computational Toxicology, and he's also the chair of Crop Life's Europe specialty group. It's a mouthful, on QSAR. And actually, he joined the BASF in 2010 as a regulatory toxicologist for plant protection chemicals, with a special focus on high content technologies and also elucidation of mode of action, which is, by the way, also my personal interest. And as part of these activities, he's a specialist in quantitative structure-activity relationships, so QSAR and read-across, which actually also mark the topic of today's webinar. So with this, I'm very happy to give the screen to Markus. So Markus, please, up to you.
Yeah, thank you, Mathieu, for this very kind introduction and for giving me the chance to present here about how we in pesticide industry are using QSAR and read-across in the assessment of our protection of our products. So what is QSAR and what's read-across? Yeah, so the underlying principle of QSAR is that we can infer or glean biological and chemical properties of a compound via looking at its 2D and 3D structures. And this has 2 underlying principles. The first one is the principle of similarity, indicating that similar compounds share similar biological activity. The second one is that functional groups confer chemical properties. Yeah, for example, you're adding a carbonyl group, yeah, or a carboxylic acid group, so you're increasing the hydrophilicity of the compound, increasing then excretion from the system. In a read-across, you are trying to take data from similar compounds and try to fill data gaps, yeah, and infer then physchem and biological properties for your query compound. And a QSAR is in a sense nothing more than using cheminformatics methodologies and statistical methods to assist in read across. Now, the problem we are having here is that we have a couple of different pillars of methodologies, and we want to use them to infer data for human relevance. On the one hand, we have our in vivo test systems, which are on the one hand very powerful because they are complex and multi-organ systems, and they contain the complete capabilities of metabolism and ADME, excretion, and so on and so on. On the other hand, they have a rather low throughput. They require a lot of material, especially if you go to higher tier assays like cancer or repro studies, and they are very expensive. I mean, just looking at a cancer study, we're talking here about 3 million euros. And of course, there are a lot of pressure from society for animal welfare. On the other hand, we have as our second pillar, our whole in vitro test system battery, where you have heard a lot about in this similar series, likely, and it's also getting more and more into focus on scientific conferences. Those in vitro methodologies have one advantage, so have a much higher throughput, you need much less material, and they're relatively cheap depending on the endpoint. However, the in vitro methodologies mostly cover single endpoints. They often have no metabolism, and definitely they are lacking in excretion of the compound. Therefore, taking this together, it's always a question, how relevant are they for the in vivo situation? And therefore also, how relevant are they for the human situation? Now, in silico methodologies have one major advantage. Now, they are extremely fast, and you can run thousands of compounds in parallel, even if you're not able to synthesize them. So you just need to come up with a structure, and then in principle, you can throw that into in silico methodology and an answer. Another advantage is that you can merge data from multiple endpoints and systems. For example, you can use data from a cancer study plus data from in vitro systems and try to glean information. However, we also here are facing a number of severe disadvantages and challenges, namely that the reliability of any given in vitro system is limited to either the level of complexity that we can model and to the number of endpoints, where we only currently have a very small number of endpoints where at least global silico methodologies are working. The second major problem is that this in silico methodologies need to be really fit for the chemical domain that you want to investigate. Saying that, build a model which is looking at the chemistry of a real hydrocarbon receptor ligand. This will likely not be fit for purpose for a model looking at more soluble compounds. Now, we in the pesticide industry are using QSAR and in silico methodologies for two kinds of activities. First of all, in research. Here we can use them as a help for prioritization of adequate candidates to be developed. Yeah, and here we can use them to identify early watchouts like genotoxicity, persistence, or bioaccumulation. We can do that on a more global perspective, looking at a very broad area of chemistry, but also for some of the chemistry where we know a lot about, we can also go into a targeted read across for higher tier endpoints. But that's the focus of my presentation today. Today, I would like to talk about how we use in silico methodologies for the evaluation of impurities and residues, which are a data requirement in our legislation. And here, our initial target is to look at genotoxic potential, where we are looking for aims mutagenicity and chromosome damage and anagenicity via the in vitro and in vivo MNT. However, more and more authorities are also asking us the question, can you model and can you provide us some read across for higher tier endpoints? And I would like to show you as a part of my presentation that this is potentially possible for some of our chemistry, but for many, we are still in very infancy of the methods. They're not fit for purpose yet. Saying that, this is our regulatory question, namely that our legislation asked us or tasked us with a toxicological evaluation of plant and livestock residues. So everything which is ending up in a dietary matrix and where the consumer may have exposure to. And here we have a legal framework setting an EFSA guidance document from 2016. However, this was not accepted by the EU Commission, and therefore the development of a guidance has been moved to OECD, and we're expecting a new draft guidance coming up end of the year, then open for commenting. So currently, we have an unclear regulatory situation. On one hand, the law says we have to do something, but we have no clear legal guidance. So we have to submit something, and we are following, in a sense, what's laid down in this EFSA guidance proposal, namely doing a combination of the evaluation of toxicological properties with a dietary risk assessment. And we do that in 4 steps. First, we do an assessment of the genotox potential for each and every compound where we have a structure. Then we group the compounds into by chemical elements or by metabolism, perform a dietary risk assessment for the group, and then, depending on the exposure, evaluate genotoxicity. And here we can partially also use the threshold of toxicological concern. To illustrate this a bit more, we are talking about that we have a large number of metabolites. This can sometimes go into the area of a few hundreds, but for most of them, we have virtually no exposure. If we have a high exposure, then of course we have to make a thorough evaluation. We have to derive points of departure for risk assessment and also identify the hazard potential. If we have lower exposure, we can also rely on the TTC concept. And here there are 2 thresholds, one for chroma class 3, so everything containing a heterocycle or reactive functional group, and another one for neurotoxicity, whether the compound has a carbamate or organophosphate structure element which could cause neurotoxicity. If we are below that, our main focus is genotoxicity. And here we are also having 2 situations. Either we can be below the threshold of toxicological concern, and if we can show that our database is robust enough that we can exclude high exposure, then that is it. We don't need to go into detail. Now, mostly we are not in this situation because our exposure assessment is usually a bit weak. And therefore, we are typically ending up in a situation where we have to evaluate all those metabolites here or residues. And we do that by a mixture of testing, QSAR, and read-across. So let's take a look at that. The testing would require us to do an Ames test and an in vitro MNT for a candidate. Now, if we do that for a large number of compounds, we are very easily going into capacity restrictions, because here we are talking about an investment of 120 to 150,000 euro per compound. And that's not because those tests are very expensive. I mean, Ames test is somewhere around 1,500 euro. But we also have to synthesize the stuff. We have to develop analytics. We have to do GLP testing and potentially in vivo follow-up if anything of this here is positive in an in vitro system. Luckily, our regulation allows us to do a prioritization of the testing. And therefore, we are grouping metabolites and we are looking for reactive structural elements. So we do that as an initial screen. And we can, of course, for example, use structure elements or profilers as laid down in the OECD toolbox. Then the next step would be to look at all those compounds, whether we have an alert for genotoxicity. And here we are following in principle the principles laid down in the ICHM-7 guideline for pharmaceutical impurities. And we perform our assessment based on a rule-based SAR system and a statistical QSAR system. So a knowledge base or expert system is in a sense nothing more than that we try to capture expert knowledge and transfer this expert knowledge into a computer system. So what we're having here is a qualitative interaction between chemical structure and biological activity. So we have a structure activity relationship model. And for example, here we are coding all kinds of chemical rules, knowing that, for example, this structural element here, if I have that, I have a high likelihood to form DNA adducts. Now, what you're doing is you are bringing those rules in a knowledge database, and then you use a computer program to actually look for those structural elements, and then you can do further evaluation. This is a very powerful approach, and this is, for example, encoded into direct, toxtree, or some of the other commercial systems. Now, the other, and here we're really talking about a quantitative structure-activity relationship model, is a statistical evaluation. So you're trying to look for a statistical correlation between a structure element and biological activity. This is one major advantage. Yeah? You only need a database of chemicals with known experimental activity data. So in theory, it's possible to derive a statistic model for each and every endpoint where you have adequate data. So you don't need to have too much knowledge of the underlying molecular mechanisms. Therefore, this is also a very popular approach. And for example, it's implemented in the Vega system, in QSAR or DeepScope, some of the US EPA systems, and many, many more. Now, but let's take a step back. What is actually needed to build such a QSAR? And here we are really talking about four major pillars. The one pillar is data. Then how do we describe the chemistry? How can we describe chemical similarity to find structural analogs? And then, of course, how do we model the data? So let's first start with data. Yeah, and here we are facing one of our major challenges, namely that of the so-called activity island or activity cliff. That means for the vast majority of compounds, biological activity is not given. And I would like to illustrate that by a picture of the Hawaiian island chain. Yeah. So for most of the compounds, yeah, the ocean is actually, the water level is actually the biological threshold after which you would say it's active, yes or no. So most of the compounds would be below the water level. And then you have some which are only slightly above the sea level. And then you have some where you actually very strong activity, like the volcano, or to illustrate something like a volcano or the Hawaiian Islands. Now, if we transfer that to a real life example, there's actually a very nice publication from Honma et al. From 2018, who published data or an assessment of data for 18,000 Japan registered compounds. And here we are talking about the Ames test data. Ames test has one advantage, it's very robust and has a very low false discovery rate. So now if we look at this 18,000 compounds, only 14% are positive. Yeah, and that's more or less what we are also expecting here from this example. The majority of the compounds are negative, and also from this 14,000 here, it's not whether we're talking about strong activity, but most of them only show weak activity, and we only have a small percentage which have very strong activity. The next question is, how do we get data? And of course, here we have the distinction between proprietary data within the chemical companies and then the public data set from the literature. And I would just like to point out here a few of the more popular database. Hansen is very popular. The ISSTY is very popular. And of course, the FDA CFSAN database for pharmaceutical products is also very interesting. Now, if you pull all the public data sets together, you're getting somewhere around 12,000 entries for an Ames test. However, here we also have a problem of data quality. If I have a positive call, yeah, then I can be very sure that maybe I have some real result. Yeah, and then it's independent if I have a positive in a TA98 or TA100, I would capture that both as an Ames test or an Ames 2. Now, for a negative call, you need more. Yeah, you need to be really sure that the assay is really negative and that you would also not capture something by doing an Ames test with all 5 strains. So you can only do GLP standard data sets. Another question you have to ask yourself is, how do you handle multiple assays? So how do you handle the situation if you have 12 assays, 3 are positive, the rest is negative? What are you going to use in your QSAR system? And to illustrate this problem of data quality, I would like to go into one of my compounds, my pet compounds. It's a denitramine herbicide. If I only use published information, yeah, I get one positive publication, and I can take the easy way out and say, okay, I take the worst case assumption and use this as positive compound. However, this is not really reflecting the chemical information that we have, or the data that we have. And we believe from industry that what we enter into the QSAR models should be based on a wide of evidence and also take into consideration up-to-date authority decisions, because there a full review of the data has been done. And to illustrate that, again, going from a compound, I know that for this compound class, nitrosamine impurities can be formed during synthesis. And actually, if we look at the data here, down below here, depicted, I see that for a large plethora of Ames tests, I have one, which is positive, and that can be traced back to a nitrosamine impurity, while for 5 others are negative. I also have additional evidence from mammalian in vitro systems, which are negative. I have a couple of metabolites tested, all negative, and I have other denitramine herbicides and their metabolites, which are also mostly negative. So if I would make a weight of evidence analysis, I would be clearly negative, and I could actually say that this positive result is likely due to an impurity. So next question is, how can I then use this information and build a model? Ideally, a model would be built on a relatively equal split between positives and negatives. Yeah, I know this is just a depiction of one of the data systems in one of the commercial models. So there you can actually here have some 6,000 active and 6,000 inactives. And then you can plot sensitivity of the model. So the true positives and the true against the specificity. And here you would have a random model. So the real data curve gives you a very nice correlation here. And you would then choose a classification threshold somewhere here as a tangent and get a nice mix between sensitivity and specificity. In this case here at a threshold of 0.5, you get a sensitivity of 90.7 and a specificity of 92.9%. Now that's an ideal situation. However, it's not really reflecting our real life. In real life, we usually have more inactives. And for example, this is also one of the commercial models. Having entered another 1,000, roughly 1,000 positives from pharma proprietary data sets and another 5,000 negatives. So we have multiple options how you can then handle this. You can either reduce this threshold, get more stringent, or you can use oversampling or undersampling technologies, or even go a step further and create multiple models with different set of chemicals, which you then combine into an assembler model. Now, for the pesticide chemical space, we are in the situation that mutagenicity is a cutoff. So we cannot sell or develop compounds which are mutagenic. And therefore, our database is extremely skewed towards negatives. So if we would like to improve the QSAR models, which are out there in either in the public domain or the commercial ones, we have to somehow bring our proprietary data into the models. If we can do that, then we can really rely on the results. If I have a positive prediction, I can either test or trust in the system. If I have a negative one, and if it's in domain, so in the applicability space of the model, I would say this is a valid prediction, and I can potentially wave testing. There are 2 ways how we can do that. The first one is that we can do data sharing. Yeah? And therefore, we would need to bring together different stakeholders, industry, and here we're really talking all relevant companies. And for example, in PropLife Europe, we have a tentative yes, that's something we want to do, but it requires a lot of resources. And we mostly would go with submission data, so data which has already been evaluated by authorities. Of course, we also need the QSAR vendors, and we need to buy in by regulatory authorities. The second, also very elegant approach is collaborative modeling. So each and every company would then be able to bring their proprietary data into a model, but the data would remain confidential. And that's, for example, done in the direct knowledge base or in several of the commercial statistical models. But it's difficult for regulatory acceptance, because then the regulators don't know whether your model is based, or the data you base your model on is actually correct, yes or no. Another major problem is IP protection. Yeah, and we need to be a bit careful here, because we are running also into the problem of antitrust regulation. If, for example, the major companies like Bayer, Corteva, BASF, Sumitomo would share the data and then limit the access to the models to these companies, you're inferring a monopoly. Yeah? And that is something which is very difficult under our regulations. So next topic, how do we describe the chemistry? And there are a lot of ways how you can describe chemistry. Yeah, you can, the most popular one is fragment-based. So you can devise fingerprinting methodologies, and that's usually done in the proprietary fingerprints in the typical QSAR models. You can use public fingerprints like PubChem, MAX, or circular fingerprints. You can go via atom-centered fragments or structural alerts. Another option is that you can use larger scaffolds or chemical force, like the beams in Mercoscope, that's very prominent in substance development, or you can go for smarts, which I'll introduce a bit more in detail later. You can use Swiss-chem properties, 3D structure, metabolism, biological activity, whatever you want, if it's feeding into your model. So let me first describe a bit about fingerprints. So a fingerprint is nothing more than trying to code your chemistry into a bit string. And what's usually done here is that you have a number of predefined structural fragments, and so you then simply count or mark whether a fragment or one chemical group here is present, yes or no. For example, here you have a secondary aromatic amine, it's present here. You have a nitro group which is present here, but not here. Then I have a couple of things like here, the peptide group and the backbone here, which are shared between the two molecules. And here I have a halogen, which is present here, but not here. There are also additions to those fingerprintings, like taking the number of elements into consideration or the positions. Yeah, and those fingerprints are very frequently used in QSAR and machine learning algorithms. However, fingerprint technology also has some major disadvantage, namely that you do not capture metabolism or you do not capture really pharmacophore information, whether a pharmacophore is really driving your separation. Something which is also very important to understand is structure alerts. The structure alert is, as indicated in the beginning, either expert knowledge dependent or it's based on the statistical ratio between the positive and negatives in your training data set for a given fragment. Here I have two examples. Again, going for my dinitroaniline. Yeah, I have here this structure. I have two phenol nitro groups, and I look at into a typical model. Like here, the case ultra Ames model. I see that the majority of the compounds in the training database containing that element is positive. Yeah, so it's a very powerful alert. Same is true for secondary aromatic amine. Now, as indicated, my database for this compound and for the metabolites in this compound class, as well as other dinitroaniline, says yes, but it's not relevant for our compound class. So I have additional elements in my compound class which prevent this from being genotoxic. So I have to evaluate that in this case, this structure alerts here are not giving me meaningful information. My model is not fit for purpose. So what I have to do here is a targeted read across and go one step further. And one step further is, for example, the use of pharmacophores or smarts. And the basic principle here is that biological activity and metabolism in a group of chemicals are dependent on specific structural elements. Yeah, for example, in the organophosphates or carbamate group, you have strong neurotoxic activity based on inhibition of the acetylcholine esterase activity. Yeah, and you can either use a organophosphate structural motif where the S also can be an O, or you have the carbamate motif. Another example would be for triazole fungicides, where the inhibition of the aldosterol biosynthesis is mediated by the presence of here this triazole moiety together with a linker and then some phenolic side chains. So what you can do is you can capture this information into structural motifs or scaffolds. Yeah, and I would like to give you here an example for the herbicide group chloroacidamides. And what you can see here is that I can describe this whole group by either describing this part of the molecule. Yeah, I can do this scaffold here. Yeah, or I can use the lower part of the molecule as a scaffold. And by adding then some wildcards or R groups, yeah, for example, I can say here, at this R position, I can have a phenolic group with either methyl or ethyl group at this position over here, or I can have this thiophene ring with the methylation groups. Yeah, I will capture a broad range of this chemistry. Now we've done that. We've teamed up, or we, in this case, is PropLab Europe member companies. We've teamed up together with a group of Mark Cronin in the University of of Liverpool, and we have done such a smart or structure alert-based motif read across for the compound class sulfonylureas. It's a herbicide compound class, but it's also a compound class used heavily in the pharmaceutical industry. And the advantage here is that this is a very data-rich compound class, plus they all share common structural motifs like the bridge here, and they also share common metabolic pathways. In addition, we from the pesticide industry are lucky that we also have access to data from metabolites and residues, which have been tested to fulfill our data requirements. Now, what you can do is first you define smarts. Yeah, and here, just for an example, here, this is our sulfonylurea bridge. And I have defined an R group here as an aromatic carbon, and a second R group here as being either pyrimidine or triazine ring. The second one would be a cleavage of the bridge here, getting into this sulfonamide group, again, with an residue here as an aromatic carbon. I can then use such a smart to delve into databases and collect information. For example, here you can look what's the database for Ames tests. That was our training data set. That's additional data pulled from the EFSA Genotox database, and I get a total of 38 Ames test data sets containing the structural motifs, which all are negative, indicating that here I don't have a genotoxic potential for this structural motif. If I look at CA in vitro, my majority is negative, but I have a few positives in the assay system, which are then turned into negatives or no longer present if we go into the in vivo situation. And that's actually a very important, interesting point here, that here we can actually pull together data from a significant amount of in vivo assays, which allows us to also not only look for chromosome operation and Ames mutagenicity, but also we can then look into data for anagenicity, which is something not covered by the in vitro CA system. So you see that by using of smarts, you can already start performing a compound class specific read across. But be a bit careful. The same structure motifs not necessarily provide you with relevant info for other endpoints. Now, the next step we would need to do is to build confidence. Yeah, and for example, in this case, we can use metabolic information. And here again, the pesticide realm is very good or cool because we have a lot of data. We have to perform a complete set of ADME studies, including metabolism in low dose and high dose after repeated and single dose exposure. We also know what's going into bile, urine, and feces, so we can distinguish what's going to be in biological bioavailable matrices or not. And we also have information of whether we have a major metabolite or whether it's a major metabolic pathway or not. And what you can see here for the sulfonylureas, we have typical metabolic reactions where we either have cleavage of a methyl side chain, we get then hydroxylation in different positions of the molecule, we have the cleavage of the sulfonylurea bridge, and we can substantiate those metabolic side chains with smarts, thereby gaining further evidence whether this metabolic reaction causes an increase in genotoxicity. This is also something which is going to be available to the public in more detail in the future, because EFSA is now requiring mandatory submission of pesticide metabolism data via MetaPath, which is a metabolism data repository format. So next part, how do we describe chemical similarity? And here, it's usually a combination of use. So Similarity is more or less looking for analogs. And it's usually done by a combination of a fingerprint with a so-called similarity matrix. For example, you can go with Tanimoto-Jacquard coefficient, you can use dice or cosine. It doesn't really matter as long as you take a consistent approach. This is just for shits and giggles that you have the formula for Tanimoto. And what you're looking here is really whether you can distinguish between the overlap and the differentiation patterns. Yeah, so you're taking these two fingerprints and look where I have similarities and where do I have differences. And then you get a similarity coefficient or value, which is often cited in the literature of a value of 0.7 being similar enough. This is very subjective. Be very careful of using any of those thresholds. And I would like to give you an example, again, going back to my pet here in the denitrile herbicide class. I would like to show you how the different combination of fingerprints can really skew your similarity cutoffs. So here I've used three very popular fingerprints, the max keys and the PubChem fingerprint and the circular fingerprint. And if I then calculate the similarity across this compound class here, I see that penimethylene is very similar to butyrylene. Butyrylene is this guy here, penimethylene is this guy. And you see here they both have a halogen, have a typical alkane side chain in this position. Now, the other ones either have a trifluoromethyl group here or have an SO2 NH2 group. And they cluster together, but are dissimilar from penimethylene or butyrylene based on the max fingerprint. If I use the PubChem fingerprint and take the 0.7 threshold, all would be considered suitable read-across candidates and fit for purpose. If I take a circular fingerprint, none of those, not even penimethylene and butyrylene, would fall together. So thresholds are really depending on the combination of fingerprint and similarity matrix. And please make sure that you understand what you're doing. Again, here we can use the alternative, namely we can use smarts. And here I can, for example, build this scaffold. Here is present in all of those compounds here. And I can then define different R groups. For example, here for I1, I can then choose that I have either a trifluoromethyl group or an SO2 NH2 group to capture the upper part or those guys here, protein and oryzalene, or I can go with a simple alkane chain, capturing penimethylene and butyrylene. Now, if I have the more or less final side chains, I block oxidation, and therefore I drive my metabolism into a certain direction. With penimethylene and butyrylene, where I have an alkyl side chain, this is becoming a primary metabolic site. You have oxidation first to a hydroxyl and then to an acid, which enables a much faster clearance than if I have a trifluoromethyl group or an SO2 HN2 group. Here I have my secondary aromatic amine, and irrespective of how they look like, I usually have hydroxylation in the better position, which in combination with a reduction of the nitro group here can result in the formation of a benzimidazole moiety. And this is a very predominant metabolic pathway in the liver of compounds containing an R group of CF3 and SO2NH2. And interestingly, this seems to be one step which is required for liver tumor formation. It's also a step which is very prominent if we are looking into the liver of goats or ruminants, which have a more reductive metabolic pathway. For penimethylene or butyrylene, this is a minor pathway. It's still major in the liver, but it's looking at the overall balance, it's a minor pathway. And nicely, those compounds do not have liver tumors. So you can directly use whatever you use for similarity assessment to infer biological activity. Now, if we do that for the denitrile and then again use this to pull data, we see that we are usually negative for enzyme mutagenicity. In contrary to our structural alerts approach, we are positive for in vitro chromosome operation and MNT. We are negative for mammalian gene mutation, and we are negative for chromosome operation and anagenicity in vivo. If we are looking at higher tier endpoints, the picture differentiates. And here, depending on the amount of benzimidazole and blockage of clearance, we may end up in a situation where we have very strong liver induction, and then secondary to that, also mouse tumor formation, likely via a non-human relevant CAR-PXR mode of action. So I hope we could show you that with a few examples here, that you can use a lot of information in your read-across and QSAR analysis. The first one, and that's the essential one, you need to understand your data. And you need to understand whether structure alerts are fit for purpose. And then you can really go further ahead, depending on how much data you have, whether you have metabolism information, whether you can use smart or fingerprint technology. You can use everything to pull together to build confidence in a QSAR and read-across. So if we take then a look at model productivity, I hope I could show you that depending on how much data you have or how much knowledge you have, you can really start building good models. And if we transfer that to endpoints, now we are usually in the position that for simple endpoints, like an AIM, log KW, or bioavailability, you can really either have a lot of data and you have a lot of knowledge, so you can build good models. If we are looking at the more sophisticated endpoints like chromosome operation or in vitro MNT or some of the ED assays, we have a high false discovery in the system, which directly reduces the data quality and whether the data is really predicting the real situation. That's making it, of course, much more difficult. Plus, you usually have less data. Same is true if we're looking at simple endpoints like in vivo MNT, because here my false discovery rate is so low that I usually don't get positives, and then it's really difficult to evaluate your models. Now, if we are looking at highly complex, multivariable or multivariate systems like repeated dose toxicity, target organ toxicity, carcinogenesis, or death tox, we are lacking. We are lacking both on the data, amount of quality, as well as knowledge. But still, we are driven to perform that by our regulatory authorities. And to give you a flavor of how the situation looks like, I would like to go a bit into EUROTOX and DevTox. So EUROTOX and DevTox are more or less the king discipline of toxicology, yeah? Because here we have very heterogeneous test systems. We are looking at two different species, rat and rabbit. We apply the substance to parents, and they're looking at effects on fertility and offspring. We have ADME changes during pregnancy. We are looking at multiple AOPs, many of those unknown, and the rabbit, our second test system, is virtually not characterized. In addition, the test is very difficult, and you have a very complex data analysis. Plus, the test is very expensive, animal and material intensive. So it's only done for a very limited kind of chemistry, either high tonnage chemicals, pesticides, where it's mandatory, or for a limited set of pharmaceuticals, where your treatment regime could also capture pregnant women with repeated exposure. Now, saying that, we have EUROTOX as a cutoff in the EU. So a pesticide company will not generate compounds which have this as an intrinsic property, and you would screen out those substances in the screening. So we also directly have a negative bias. On the other hand, for everything where we don't have a direct data requirement, like low tonnage compounds, like food contact material, cosmetics, biologicals, we don't have data at all. So we have a very limited data pool we are looking at. Still, there are models, and the models are limited. Yeah, for example, they usually do not contain pesticides and no newer REACH data. The QSAR models do not differentiate between developmental and reproduction toxicity. And also looking at the effects, it's usually this differentiation between is positive or negative. So it does not take into consideration variation with smile formation, sexual delayed maturation, or fertility effects. Also, to have a higher predictivity, maternal toxicity is usually not taken into consideration. Now, we did a little exercise and tried to benchmark those prediction models against the data for pesticides. And here we collated a database for EU registered pesticides and looked at the GHS classification. We then took advantage of the available structural alert models and statistical models and did a comparison between the predicted data versus our in vivo information. So let's first take a look at our pesticide data. And here we are really talking about a full data set for 2 generation OCD 416 and both a rat and rabbit DevTox data set according to OCD 414. So again, our data is very much skewed. The majority of the data is not reproductive toxic, with a couple which have a ReproCut2, and then a limited number, mostly based on single compound classes, where we recently received classification on Repro1b. But beware a bit. This is maybe not reflecting the full picture, because the database for pesticides is very heterogeneous. We have studies performed according to the old data requirements and new guideline studies, and therefore we have not everywhere captured every endpoint. It's also that for the newer compounds, the OCD 416 captures a lot of additional endpoints like ED-related and sexual maturation-related effects, and also hormonal effects, which are not available for older compounds and for compounds which no longer have an approval in the EU, because companies will not invest any longer in them and no new data is generated. Also, here our focus was based on ECHA classification, and the classification standards have changed over time. And for compounds which have not been reviewed by ECHA in the last years, you have certainly completely different standards than we would have today. Nonetheless, I mean, it's as good as we can get, and this is just a depiction of the model in performance. I mean, this is a ROC curve I've introduced before. So ideally, you would expect the curve of sensitivity versus specificity versus sensitivity somewhere in this area. Now, what you can see is this very much not the case. So let me first guide you through this. This is the different models coded via colors. And here, that's the more important information actually, it's the number of pesticides actually covered by the predictions. So only for most of the models, we have only for a very limited coverage of the pesticide space. For example, here for this lead scope repro female model, we have a good specificity, but we only have 30% coverage. So the majority of the pesticides cannot be predicted at all. For others, we have higher coverage, but then we either have less specificity and sensitivity, so the models are clearly not fit for purpose. So there's urgently need to improve these models if we at any time in the future would want to use those models. And there are a couple of solutions. First of all, we have to supply more and high quality data, especially the company-owned data. We really have to differentiate the endpoints. We have to investigate what has been tested in which assay system, and then make this data available to the model builders. We can include AOP information. We can include more knowledge about the mechanisms of reproductive toxicity, both for developmental as well as then for example, sexual maturation. So at the moment, we are not there, but we are in the process of developing into that direction. With that said, I would like to come to the end of my presentation, showing that we can pull together a lot of weight of evidence into good QSAR and read across. And if you have sufficient confidence, this is actually already sufficient to waive testing or to do selected testing. However, we need an urgent exchange between all the stakeholders. So we need industry on the table, we need academia on the table, QSAR vendors and the regulatory authorities to really bring together to define what's needed to make a QSAR model fit for purpose. How can we bring in the data, and also for read across how to build confidence and what kind of methodology is acceptable to regulatory authorities in order to accept a read across and wave testing. With this, I would like to come to the acknowledgements. I would like to thank everybody involved from CropLife Europe for the sulfonylurea work stream, also the people from the University of Liverpool, and my PhD student Anastasia Weyrich and her co-supervisor Thomas Hoffman, as well as the people from preclinical science for the work on the EUROTOX models, And of course, thanks people from the different QSAR vendors for their help and assistance with all the evaluations. With that, I would like to thank you for your attention, and I'm happy to take any questions.
Thank you so much, Markus. This was a very, very nice and overview and a very complete overview. So thank you so much for that. I didn't see any questions yet in the chat. So please, people, if you have a question, either insert it into the chat or you can speak up. Maybe I can take off with a question from my side. So Markus, you mentioned at the end that these models are not fully ready yet, let's say for regulatory testing purposes, or let's say for hazard identification. But even in the case that hopefully in a couple of years, they will be more eligible for using in a risk assessment and probably in a kind of a probabilistic risk assessment approach. Do you think that for hazard identification, these models like QSAR and so on and so forth would be able to be used, or will we be able to use them as standalone tools, or would you advise to always combine them with other approaches, for example, in vitro testing?
I have a very clear position on that, Mathieu, and that's saying that we are clearly by far not there. I mean, if you're looking at an in vivo situation, you're talking about 50 organs, and you need to have a lot of information both on the underlying mechanisms, which we don't know, and also of the data. Also, if you're looking at a global perspective of all the chemistry which is out there, we certainly will never have enough data. So we can use the QSAR and in vitro systems to drive information and then help us do selected testing. But I believe, especially for high tier endpoints, we always need proof from an animal.
Yeah, I actually agree on this. So thank you for confirming. And then another more technical, well, a more specific question, I would say, from my side. So I'm a pharmacist by education, so I always pay special attention to, any, let's say, pharmaceutical compound. And actually, the example you gave with the pesticides, so the sulfonylurea compounds, well, probably you know this, but these are also actually used for treating diabetes. So my question to you is, to what extent with all of the tools that you now nicely presented, would this also be applicable to other sectors, so the pharmaceutical sector? And if already done, so for example, in the case of the sulfonylurea, I can imagine that being drugs that are on the market, that there is a wealth of data already out there. I mean, could this also help to further optimize these models?
Oh yes, Mathieu, definitely. I mean, we as BASF are also part of the so-called e-Transafe consortium, a large pharma consortium trying to bring together preclinical data and in silico modeling, and then try to link it to clinical data. Now, we from pesticides don't have human data, so for us it's extremely interesting to get the human data. But the major strength of in silico methodologies is that you can pull together data from different sources. And as the pharmaceutical industry has all the human data, we would be in the future potentially be in the position to also pull in data from clinics, to also look whether you have kind of information that could say, okay, here I have a metabolic pathway, for example, which is relevant for an in vitro system or for a mammalian system, but it's not relevant for the human situation, and vice versa. Or you have specific target organ toxicities that you cannot capture, and then you can really use the data and decide whether you need to do a dedicated testing.
Okay, thank you. And then I see that unfortunately there's still no questions. I think this might be a good sign because everything probably will have been very clear then, but a personal question still from my side. So I'm currently coordinating a European project called EUROTOX. Since 1 year, it's a quite big consortium in which we try to set up approaches for animal-free toxicity testing or safety testing, I should say, of chemicals. So it's not only about hazard identification, but we try to do full-blown risk assessment, so-called probabilistic risk assessment. Now, a very important tool in that approach is artificial intelligence. So in that respect, I mean, QSAR can also be considered as artificial intelligence, but you being the specialist in computational toxicology, what would be your opinion on, like artificial intelligence in the field of toxicology and risk assessment? So how do you see this evolving? Do you see a role for this?
Yeah, thank you, Matthew, for the question. It's very important. We have to be a bit cautious here. On the one hand, I mean, if we are looking at artificial intelligence and what's been promised for other fields, we're talking on really big data. Yeah. And for toxicology, we are talking on data for a couple of thousand compounds, where we actually have data, and therefore we are rather limited. Then we also have a very broad aspects of endpoints that we want to look at. Now that is not a prohibition, yeah, and the field is developing, and also we are getting a lot of developing in oversampling, undersampling, like what I mentioned. So we may have a good information for some data sets or some compound classes. And it's actually a field where we have a lot of research, especially also from the pharmaceutical sector. To me personally, I believe we need to first clean up our data, to really understand what kind of data do we have from the in vivo situation, what has been tested where. And if we have done that, there are a lot of efforts going to that direction, like standardization of studies, and really getting good protocols for study reporting. We will enable our data to be used in artificial intelligence research. Once we are there, of course, we can use it.
Yeah.
One more comment to what you said, probabilistic risk assessment. I really like this, and I believe there's also one of our strengths here, that we can really get a better feeling where we can empower risk assessment versus hazard assessment. How we can, for example, use the data that we have in benchmark dose modeling, define the variability in the assay systems that we need to take into consideration, or actually go a bit into the direction for which endpoint can we play around with our safety factors. Is it human relevant, yes or no? And there is a strength in the system that you can pull together all the data.
Yeah, exactly. This is fully, I would say, echoing the concept of the EUROTOX project. So I'm happy that we are on the same page. And I'm not a modeler myself. It's really not my expertise in silico toxicologist. I'm more at the in vitro side, let's say. But this is indeed very much in line with what I keep on hearing from the in silico modelers from the consortium, like, well, with all due respect, and to put it in a bit of a blunt way, but there's a lot of crap out there. So we need to curate and to clean up the data before we fully can exploit, the power of artificial intelligence. So it's good to hear that this is also being, confirmed by you. Okay, I have a look here at the time, and I see it's 1 o'clock, at least in Central Europe. So we didn't get any questions from the audience, but if that would be okay for you, Markus, so if people would still have, like a question or so, I would like to ask you it personally, so maybe they can mail you if that is okay. Okay, thank you so much. And again, this webinar also has been recorded. It will be uploaded to the EUROTOX website, specifically on the specialty section, part of EUROTOX. And with this, I only remain to thank you, of course, first of all, Markus, for this excellent presentation, and which is very relevant, of course, for In2Tox. Then I also would like to thank the audience for, bearing with us, I would say. And of course, I also would like to thank Elaine. You didn't see her, but she's here. I mean, she's actually organizing all of this, so taking care of the registration, also sending out the invitation, which is much appreciated, of course. So with this, have a good remainder of the day and hope to see you soon, maybe in Maastricht during the EUROTOXicity meeting. Bye, everyone.
Bye.